Sure.
So we are now recording, and thank you very much for agreeing to give a Build a Cell seminar first in 2020, and it's all yours.
Great, thank you. Let me share the screen. Does it look all right to you?
Yep, it looks perfect.
Great. Okay. So first, I wish everybody a happy New Year, and I should thank Kate for inviting me to give the Build a Cell talk. And my name is Bo Zheng, as you can see here. And my talk's title is Hydrogel-based artificial cell. So before I share my research work, I would like to introduce myself a little bit about my background. I obtained my bachelor and PhD degrees both in chemistry and also did my postdoc research in chemistry, more especially in developing droplet microfluidic technologies, just as shown in this kind of image. In this microfluidics, we can produce uniform droplets with the dimension of around tens to hundreds of micrometers. So this kind of droplets provide an ideal platform for microbiochemical analysis. For example, high throughput screening, enzymatic assay, digital PCR, and single cell analysis. I started my independent research at the chemistry department of the Chinese University of Hong Kong until last year. And so here I continue developing different microfluidic technologies for biochemical analysis. So recently I moved to Shenzhen Bay Laboratory, which is a newly established research institute in Shenzhen, focusing on biomedicine research. So this Shenzhen and Hong Kong are essentially twin cities, just like Minneapolis and St. Paul. They are separated just by a river. So the droplets produced in droplet microfluidics, they have similar size of mammal cells, and artificial cells have always been a fascinating topic to me. So in recent years, I started applying droplet microfluidics to the preparation and study of artificial cells, which is what I'm going to talk about here today. And I'll ask, this is probably very easy for all of us, and so I will quickly talk about this. So the concept of artificial cells starts when people prepare emulsion system made of enzymes and proteins encapsulated in membrane. So, and different types of artificial cells are constructed as cell mimicking systems to study and understand like properties of natural cells, and also to investigate the dynamics of cells without the complexity of natural cells. People can also explore new applications in synthetic biology, metabolic engineering, and biosensors by using artificial cells. So to construct or to build an artificial cell, we have the top-down and a bottom-up approach, as shown here. So for example, in the top-down approach, people take away or modify part of the genomes of the living cell, so that the complexity and will be reduced. The best known example is the synthetic bacterial cell from Craig Venter Institute. And on the other hand, the bottom-up approach assembles different biological molecules, such as the nucleic acids, proteins, and the lipid molecules, to form a structure that reproduces the organization and the functions of living cells. So for a chemist like me, the bottom-up approach would make more sense, and is something more attractive and logical for building and studying artificial cells. Of course, there are many other chemists and also biologists who also think this way. Many different types of artificial cells have been constructed from bottom up. We can roughly divide those structures into two groups. And first, those microencapsulation systems, such as fatty acid vesicles, phospholipid vesicles, polymersome, proteinsome, and membrane encapsulated coasphates. And there are also the second group, which are non-encapsulation system, such as the coasphates, porous particles, and hydrogel particles. So in natural cells, all the functions are carried out by proteins, from signaling and catalyzing reactions. To have the same functions, the artificial cells usually also need to begin with protein synthesis. And this is usually achieved by using cell lysate from, for example, E. coli, wheat embryo, or rabbit red blood cells. So in artificial cells, the protein synthesis can be carried out in one of the two modes, in batch mode or in the continuous mode. So batch mode is easy to understand. For the continuous mode, this is especially attractive, and people use, in this mode, people usually use some kind of a membrane, either the membrane used to encapsulate the artificial cell, or an extra membrane to maintain the large protein molecules for transcription translation, while allow the small molecules to freely diffuse in and out of the artificial cells. However, the artificial cells I mentioned in the previous slide, in this slide, they usually present a few common problems. First, the cell lysate they often contain inhibitive or digestion factors. And secondly, the cell-free systems in the artificial cells, they lack the control of mass exchange with the surroundings. Consequently, the protein synthesis would not last very long in this kind of artificial cells. Usually for only a few to tens of hours. So to solve this, to solve the first problem, we can utilize a protein synthesis system called pure system. So this is a system developed by Professor Ueda from University of Tokyo in 2000, and it is a protein synthesizing system reconstituted from the recombinant tagged protein factors, purified to homogeneity. So the pure system contains 36 enzymes, as listed here, that are involved in transcription and translation, and also the highly purified 70S ribosome. The advantages of pure system is that it has no proteases or ribonucleases, and its pure system is able to produce protein with high efficiency. Another prominent feature of the pure system is that all the enzymes and factors are heat-stacked. So the protein product can be easily purified by using affinity chromatography. To solve the second problem, that is to control the mass exchange, our strategy is to immobilize the pure system or the cell lysate on the polymer backbone of the hydrogel microparticles to form artificial cells. So in this way, the artificial cells remain an open system, allowing continuous uptake of nutrients and the release of protein and waste products. In the meanwhile, because the pure system or the transcription translation system in the cell lysate is immobilized, we would expect that there would be no diffusive loss of the protein component. of the transcription translation system during the protein synthesis. First, we synthesize the hydrogel, which is a polyacrylamide hydrogel, by following the conventional photon-initiated polymerization method. So in the reaction, we added acrylic acid, NHS ester. So the resulting hydrogel backbone would contain NHS ester function groups on the polymer chain. We can produce uniform hydrogel microparticles by first generating uniform water droplets in oil in microfluidic channels. The water droplets contain the monomers and initiator and cross-linker for the polymerization reaction. And when the droplets were exposed to UV illumination in the downstream, polymerization occurred to convert the droplets into polyacrylamide hydrogel microparticles. So to prepare the artificial cells, we first freeze-dried the hydrogel particles and then rehydrate the particles by incubation with the mixture of either the pure system or the E. coli S30 extract, as well as the DNA template. So the proteins in this way, the protein from the pure system or the E. coli extract would be immobilized during the rehydration process, converting the hydrogel particle into artificial cell. So we tried three different approaches to incorporate the different function groups on the polymer backbone, including nickel NTA, anti-histag aptamer, and PEG and PEG NHS function groups. So the first two types of hydrogel were used to immobilize the pure system through the interaction between the histag and the nickel NTA or the histag with the anti-histag aptamer. So in the third type of hydrogel, the transcription translation system from the cell extract of E. coli can be immobilized by forming covalent bond between the amino function group of the proteins and the NHS function groups of the hydrogel polymer backbone. So after the preparation of the artificial cells using hydrogel particles, we first tested the protein synthesis in the artificial cell. So the figure here shows the M-cherry protein synthesized by the artificial cells under the fluorescence microscope or the bright field. This is a bright field image of the same artificial cell. We also connected and analyzed the surrounding solution by SDS page. So the gel electrophoresis results show that the M-cherry was released to the surrounding solution after the protein synthesis in the artificial cell. So this result is consistent with the control experiment. Using solution phase, so 3 and 4 is using the artificial cell, and name 1 and 2 are the result from the solution phase. The gel electrophoresis experiment also confirmed that only M-cherry was released to the surrounding solution. If we look at the result from lane 3 and 4. So, and we did not observe any trace of protein factors or ribosome in the surrounding solution, confirming that all the proteins and the ribosome were immobilized in the artificial cells. So after we confirmed the capability of the protein synthesis, we went on testing the active lifetime of the artificial cells. So for this testing, we placed the artificial cells in a microchamber, and we flew the feeding buffer through a side microchannel. The nutrient would continuously diffuse into the microchamber, and also the product, the protein product and the waste product will diffuse out into the microchannel. We found that with continuous supply of the nutrient and energy, the protein synthesis in the artificial cell remained stable for over 28 days. We were quite surprised by such a long active lifetime in protein synthesis. We believe that the immobilization of the enzymes on the hydrogel backbone played a very important role in terms of protecting the enzymes from degradation during the protein synthesis. We also compare the efficiency of the protein synthesis in the artificial cells. We added in this experiment, we added the same amount of pure system in two and also feeding buffer in two systems. In the first one, all the pure system proteins and factors were immobilized in this one, or immobilized in the artificial cells. In this control system, we have everything in a homogeneous solution without the hydrogel particles. So, both are performed in batch condition. And after 4 hours protein synthesis, the artificial cell system produced 197 microgram per ml EGFP. And in the control solution phase, we have 87 microgram per ml EGFP. So we think there are two factors contributing to the higher efficiency of the hydrogel particles. First is by immobilizing all the enzymes and the factors in the pure system, in the small volume of the hydrogel particles, the local concentration of those enzymes and factors were greatly enhanced. And secondly, the flexible hydrogel backbone in the hydrogel facilitated the interactions among different enzymes and factors in the pure system during the protein synthesis. We also tried synthesizing a protein, urokinase. Urokinase is a protease that catalyzes the conversion of plasminogen and to plasmin. So urokinase contains 6 disulfide bonds, so making the synthesis more difficult than the previous model proteins such as GFP and or anchari. So to synthesize urokinase in hydrogel-based artificial cells, we checked that we looked into literature and revised and adapted the process. So here we immobilized the protein disulfide isomerase as well as the chaperones together with the transcription translation system in the artificial cells. We also added oxidized and reduced glutathione in the feeding buffer. So in this way, we were able to produce urokinase in the output solution. The Western blot analysis of the output solution showed the presence of the urokinase product from the hydrogel-based artificial cells, which is in name 1. So all the other names are a control experiment. I will just skip the details here. The Western blot analysis provides the yield of active urokinase synthesized for 15 hours in the control solution, and also in artificial cells made of PEG-NHS modified hydrogel, and also in the artificial cells as a control made of unmodified hydrogel. So under the batch condition, the artificial cells made of the PEG-NHS modified hydrogel had the highest yield. We also performed the continuous synthesis of urokinase in the hydrogel-based artificial cells for 30 days. 2 types of artificial cells were tested here, just as in this case. So we can see that the artificial cells made of PEG-NHS modified hydrogel has presented the yield in the range between 30 and 40 microgram per ml, and the yield in the artificial cell made of unmodified hydrogel quickly dropped to a very low level. So this result also, both results suggest that the PEG-NHS function group in the hydrogel is very important or is essential for the efficient protein synthesis. So the high efficiency of the protein synthesis and also the long lifetime in the artificial cells allow us to further regulate, to study the regulation of protein synthesis. So here we introduced the classic LAC repressor encoded by the LAC gene. And the T7 RNA polymers in the pure system will especially recognize the T7 promoter, and then the mCherry gene contains the LAC operator sequence, which can bind to the LAC protein. So the binding would prevent the T7 RNA polymerase binding into the T7 promoter, so the mCherry gene will be prevented. And if we add IPTG into the solution, into the filling buffer, then IPTG is a small molecule that can bind to the LAC protein. So when there is IPTG in the filling buffer, the LAC would be released from this LAC operator sequence. Therefore, the mCherry synthesis can be promoted. So in the plot on the right side, we can see that when there is no IPTG presented in the filling buffer, the fluorescence signal from the mCherry is low, indicating the mCherry synthesis is suppressed by the LAC protein. When we gradually increase the concentration of IPTG in the filling buffer from 1 millimolar to 15 millimolar, we can see that slowly the synthesis of mCherry gradually increase as a result. So we can tell that in this case, we can regulate the mCherry synthesis by controlling the concentration of IPTG in the filling buffer. So on the other hand, we can also see this as this is indicates that we can also use the artificial cells as a kind of biosensor. In this particular case, to detect the concentration of IPTG. Although IPTG is not a very interesting target, but the point is that if we can use appropriate gene design, we should be able to detect other more important target molecules through the expression regulation. And next, we designed a very simple genetic oscillator. So in this genetic oscillator, the protein LAC-I and EGFP were synthesized simultaneously. Initially, the concentration of both LAC-I and the EGFP are at 0, and they start to increase. The accumulation of the LAC-I gradually inhibited the synthesis of EGFP and the LAC-I itself, which provide the negative feedback of the gene circuit. The accumulated LAC-I and EGFP gradually diffused out of the artificial cell, which caused the decrease of the LAC-I and the EGFP concentration in the artificial cell. So therefore, once the LAC-I and EGFP concentration decreased to a certain level, then the LAC-I and the EGFP synthesis rate would gradually pick up again. So the concentration would start increasing as a result. And this would cause the oscillation of the synthesis of both EGFP and the LAC-I, which can be observed by observing the fluorescence intensity from the artificial cell. So later we constructed a more complex but more stable genetic oscillator. In this genetic oscillator, the inhibitor removal was achieved not by diffusion, but through the enzymatic degradation. So we used the artificial cell, the hydrogel artificial cell, to host the sigma 28 oscillator, which is with an activator repressor motif, and this is based on the genetic elements from the equalized cell-free toolbox 2.0 developed by Professor Neuro. And in this genetic oscillator, the sigma 28 was synthesized, and it promotes the synthesis of C1 SSRA protein. The accumulation of C1 SSRA would inhibit the synthesis of sigma 28 and also inhibit the synthesis of DGFP. So SSRA is a tag. This tag would facilitate the degradation of C1 protein by the CLP-XP protease, which exists in, which we already have it in the artificial cell. So when the concentration of the C1 SSRA started decrease through the enzymatic degradation, the sigma 28 synthesis would start again, which also promote the synthesis of the C1 SSRA itself, as well as the synthesis of DGFP. So as a result, again, we would observe this oscillation of the synthesis of both the protein DGFP and also the sigma 28 and the C1 SSRA. The advantage or the of this second genetic oscillator over the first one is that it is more stable and it has a much larger concentration window for us to observe the oscillation in the artificial cells. So from these examples of the genetic oscillators hosted by the artificial cells, as well as a protein synthesis works, we demonstrated that the hydrogel-based artificial cells provide a very useful platform for the research of both cell-free protein synthesis and cell-free synthetic biology. We expect that the hydrogel-based artificial cells can also find, will also find applications in several other research fields. For example, the metabolic engineering, biosensors, cell-cell communication, and the connective behavior of active matter. So to summarize, we successfully constructed several types of hydrogel-based artificial cells, and those artificial cells support very long-lived and efficient protein synthesis, and they also support the genetic programming. And this type of artificial cells provide a platform for cell-free synthetic biology. So finally, I would like to thank my students who have spent many hours working on the projects. And I also wish to thank the financial support by the Chinese University of Hong Kong, as well as the RGC of Hong Kong. So thank you for your time.
Thank you very much. This was a fascinating talk. There is a lot of questions in chat. Can you see chat?
Yes. Right. So, oh wow. Yes, there are many questions. Should I just go through each one?
Yes, if you could read a question before you answer it, that's going to be for the people that watch the recording.
Sure. So the first question is by Andrew Parkinson. What is the hydrogel's tolerance for divalent cations? could you increase, for example, the magnesium concentration of the transcription translation in hydrogel? In terms of divalent cations, we didn't specifically test the concentrations. I think we when we performed the protein synthesis in terms of the feeding buffer, we were following the suggested concentration in terms of ingredient. We were following the suggestion because the pure system was a commercial product, so we were following the suggestion provided by the company who was providing the pure system. And another thing is, in terms of the divalent cations, we did have the concern when we first, because in the first hydrogel-based artificial cells, we were using nickel NTA system. So we were a bit concerned about the nickel ions, the effect of nickel ions on protein synthesis. So that's the reason we decided using later we're using the anti-histag aptamer instead of nickel NTA. And in the aptamer case, we were concerned about the effect of ions, and we studied. If I remember correctly, we studied a few different, compared different cations on the aptamer binding affinity towards histag in that case, but not especially. We tried a few different types of cations, but my impression, because I don't remember the detailed number, but my impression is that the hydrogel has a good tolerance in terms of the cations, different types of cations, such as magnesium, calcium, this type of cations. And as long as the protein synthesis can proceed, and what I mean is there exists a reasonably broad range of a reasonable window for, in terms of the concentration of the cations for the protein synthesis to proceed. And the second question is by Harold Muro. Have you ever tried any other cell-free protein synthesis, or pure system is the only thing that works so far? We also tried, so in addition to mCherry, EGFP, Urokinase, we also tried another small peptide, another protein called collagen. It's a protein that can be used for antibacterial this kind of treatment. So naturally it cannot be synthesized in E. coli, and we were able to synthesize that protein in pure system, in the extract from E. coli. As I said, so pure system is not the only transcription translation system we used. We also used the S30 extract from E. coli. So far, that's the two transcription translation systems that we have used. The third question is, can you change the density of the hydrogel to increase or decrease the local concentration of protein synthesis components? From Petra AC. Yeah, that's a good question. We tried different densities of the hydrogel, and what we observed is that the protein synthesis is highly dependent on the density. If the hydrogel is very dense, or it is even in an intermediate density, the protein synthesis efficiency would be low. The density has to be low enough for the hydrogel to allow the diffusion of everything inside the hydrogel to be to facilitate the protein synthesis. So that's something. Actually, in the beginning, we had to try many different density of the hydrogel to find the optimal condition. Because if the density is too low, then the hydrogel would not be. It's more like a liquid instead of a solid, and so it becomes very difficult to handle. So in the end, we have to find some optimal density for both for the mechanical property and also for the optimal protein synthesis yield. The next question is from Mr. Moon. What's the biggest scale you have tried? Oh, the volume. Microfluidics would be great, but it would be fantastic if a larger volume production is possible. Yeah, that's the. So currently, this is the I would say disadvantage of using microfluidics, as you suggested, because the volume is low. Every particle has the diameter around somewhere between 10 to 50 micrometers. And the total amount is also very small. So the total yield will be limited. If we really aim for high, like large scale production, probably instead of using microfluidics, probably it will be better to use something like an emulsion to make a greater, like many more particles, many more hydrogel particles for the production of a much larger scale of artificial cells. Then, because in that case, I guess whether the artificial cells is uniform in size would no longer be important. If the production yield is a more important goal. Next question by Carrie. The oscillator experiments are very interesting. What is the time limit for the experiment? How many hours can you keep it going in the hydrogel with nutrient delivery? Yeah, I believe this is the advantage of the hydrogel-based artificial cells, because just like the protein synthesis we have demonstrated, for protein synthesis, it can last for at least 28 days, and we didn't observe the decrease of the activity for protein synthesis. So I believe the oscillator experiment can also last for many days, because we didn't observe the change of the oscillation pattern or the magnitude. But on the other hand, we didn't keep it going for like an extremely long time for many days, because it just it didn't provide any extra information in that case. But I believe it can last for a very long time, just like the case of protein synthesis, because hydrogel is an open system, and also the enzymes immobilized in the hydrogel have very long lifetime because of the protection of the hydrogel. Next question is by Mei Qi. What is the PEG modification rule which promote the cell lifespan and efficiency of protein synthesis? We didn't try many, but we tried a few different lengths of PEG. So the general observation is that longer PEG chain would be better for the protein synthesis. if we don't have PEG, or if the PEG chain is too short, the protein synthesis efficiency would be low. Presumably because the immobilized enzymes would be the movement of the enzyme immobilized would be very limited if the PEG linkage is too short. So longer PEG chain would definitely help here. We didn't try to find out what is the upper limit in terms of the PEG chain, but currently we're using PEG 2000, and that is so that kind of length already produced a satisfactory yield for us. The question from Emma Jones: can all size of molecules diffuse inside the hydrogel? Could you, for example, diffuse short oligos in or out? This would be great for environmental detection or bioreactor control. There is a limit of the molecules that can diffuse freely inside the hydrogel. We tried. I think at this moment, my impression is that it's somewhere around a few hundred, like 100 or several hundred kilodaltons of proteins. If the protein is too big, then it will not be able to diffuse easily in or out of the hydrogel. So still, on the other hand, it also depends on the hydrogel density, the cross-linking density. So if the cross-link density is high, then the protein that can diffuse freely inside the hydrogel would also have a lower threshold in terms of the molecular weight. So short oligos should have no problem because generally they have a small molecular weight. So small molecules, it should be fine. If the protein molecular weight is over 100 kilodalton, then we probably need to lower the density of the hydrogel, or we have to find other ways to facilitate the diffusion. And this question comes from Sergey Pamfer. Could this oscillator be used to drive the pulsatile synthesis and the release of drugs in situ? Probably not at the moment. I think I understand what you mean, but in terms of drug release, it will. So first of all, the hydrogel we are using is based on polyacrylamide. So this material is not biocompatible. I mean, for medical usage and this kind of in situ usage. So, and that's also another topic my lab is trying at this moment. We're trying to replace the polyacrylamide hydrogel with other more biocompatible hydrogel materials. And so in future, if the materials issue is resolved, the oscillator could be, in that case, then it could be used, has a potential, I would say, for the purpose of drug release, this kind of application. Next question is by Brendan. What is the Hydrogel 101 first step for someone who would like to get started on those experiments? What do you think is the most reliable chemistry for the reliable, stable, high yield hydrogel? Polyacrylamide is pretty standard. It is one of the easiest one to begin with. I mean, my lab is not an expertise, does not have the expertise on polymer chemistry. And we also made the hydrogel by looking through literature. And so polyacrylamide is pretty standard and mature. Another good option is probably something like PAC-DA, and as well as, and then there are some kind of for other type of hydrogels we haven't tried. We tested a few, and there are also hydrogel used in cell culture, for example. Those are also good candidates for testing. I guess it depends on the purpose. Like what are you going to use the hydrogel for? And I'm sure you will be able to find enough literature about the related hydrogel, including the synthesis and property study. The next question is from Mr. Moon again. Oscillation periods are fantastically short, probably governed by degradation only. Have you tried tuning its behaviors? We haven't done much in terms of the periodic studies. But I agree. Yeah, this is a good question. The most obvious difference between the two oscillator we developed is the constriction window. Because even if you have the genetic circuit there integrated in the artificial cell, it doesn't mean that you are going to observe oscillation just by throwing the artificial cell in a buffer solution. There is for some genetic oscillator, the requirement of the constriction of everything is much more stringent. The window is much narrower comparing to other genetic oscillator. So that's why after the first genetic oscillator, in the talk, we developed the second one by using protein degradation to control the concentration of the inhibitors. I guess I'm sure we can tune the. There are several factors parameters we can control to tune the oscillation behavior. For example, the concentrations of proteins or DNA templates, and also temperatures, as well as the hydrogel density. But we haven't done the detailed characterization. I guess it depends on what kind of insight we want to obtain by studying the genetic oscillators. So and then
so may I comment or kind of ask or a little bit more question, not more question, but comments. So the reason I asked that question is this is a fantastic system, and then also very robust kind of oscillation. This is probably not easy to achieve in bacterial or the in vivo system, but this is the possible because of using in vitro cell-free system. So, but the reason I'm kind of asking that question is if we kind of change the oscillation period, let's say increase somehow, that might also answer the previous person's kind of question about the drug release kind of periodic kind of drug, because I mean your period is around 1 hour ish. I don't remember exactly, but if you kind of increase that period, you could kind of have lots of other application. At the same time, this is the very interesting, very robust. I mean, I did never seen those kind of things before, except some recent paper. I mean, people publish. So this is fantastic. So that's kind of the reason I kind of asking the question. Also, this one, important to understand the gene kind of regulation and speed of the expression and so on. This is fantastic. I mean, for those kind of things. So that's why I'm asking that question. Also,
I see.
to answer some of the other people's question, have you kind of tried, EDC and NaH kind of chemistry that is kind of typically kind of standard in hydrogel?
It's we use the NHS. So we use the NHS system in.
Right. But you mentioned but did you use the EDC as a kind of activating molecule? Because I use polymer before to slow release some drug, and that might be one thing people could use. Somebody asked about the hydrogel 101, and also have you kind of also try the high hyaluronic acid or some biopolymer that can be good target for your the biocompatible kind of polymer, because high hyaluronic acid is very, very good. Also biocompatible.
You're right. That's true.
So anyway, so it's fantastic system. I love it actually. I love it a lot. So thank you.
Thank you. So let me continue the question. So next question is from Peter Bandel. What is the temperature stability of the hydrogel? Can you prepare hydrogel and freeze it for storage before reaction? Yeah, hydrogel itself is quite stable. It's definitely more stable than the proteins. So usually it's the enzymes immobilized that we worried about, not the hydrogel itself. One thing for longer storage, one thing we tried is that we can, after we prepared everything, and the hydrogel particle immobilization of the pure system or the cell extract in the hydrogel, and then we can freeze dry it. And in that case, they can be stored in the powder form for a very long time. We didn't test it extremely long. So far, the longest storage we tried is 6 months, and we didn't see obvious loss of activity in terms of protein synthesis. So that's the result we have so far. The last question is by Aniko X. Following Emma's comment, you could use fluorescence recovery after photo bleaching to measure the mesh size within the hydrogel. Once you know the mesh size, you can measure the diffusion constant of molecules or particles with different size. Right. Thank you. Yeah. I agree. Yeah, that's an excellent idea. Yeah. We probably should do that. Yes. Thank you for your suggestion. That's all the questions I have so far. Thank you very much. Those are all very good questions, and I think I learned a lot by discussing with you.
I learned a lot too. Thank you so much for answering all the questions. I have a couple more questions myself, but I'll follow up via email because we're running out of time already. Thank you so much again for the talk. Thank you everyone else for the discussion.
Thank you for inviting me.
And see you all next week. Bye.
Bye. Thank you. Bye-bye.